Now, I always love talking about muscle fibers, primarily because in physiology, I think we make the mistake of talking about them incorrectly. Often we like to put them specifically in groups or silos, and you see that on this screen, right? I have your type 1 fibers, your type 2A fibers, and your type 2X fibers, as if they exist as 3 different categories. But that is false. That is not how muscle fibers exist. We do tend to group them this way based on certain characteristics. Are they slow oxidative type 1 fibers? Are they fast oxidative glycolytic type 2A fibers? Or are they fast glycolytic fibers, or your type 2X fibers? And that's based on characteristics. It's also based on the size and the properties of the muscle fiber. But it's really important to point out that muscle fibers exist on a spectrum. So you could have a fiber that very much falls in between a type 1 and a type 2A fiber, or that falls very much in between a type 2A fiber and a type 2X fiber. So it's not like they all perfectly behave the same within one category. They very much exist on a spectrum, and based on training, you can have very small shifts in the behavioral properties of a given fiber type. Now, something that's interesting is that as a fiber, the characteristics are going to be largely dictated by the alpha motor neuron that innervates the fiber, and specifically how that alpha motor neuron communicates to the fiber. And we'll talk about that a little bit more, and your book does a good job of discussing that as well. But basically, how often and to what degree the neural stimulus goes to that individual muscle fiber will dictate its properties. So if you think about an oxidative fiber, for example, we use a lot of oxidative fibers in stability, right? I'm standing right now with good posture, so those muscles have to be activated pretty regularly. So it's receiving a very chronic signal of a low amplitude to the muscle, constantly sending a signal to my abdominals to hold me straight up, right? Versus if I wanted to go sprint, I'd be sending a very strong stimulus to my quadricep muscles to engage in that activity. So you can see where the way that the neuron itself would communicate with the muscle fiber might actually dictate what the behavior is for the muscle fiber instead of the other way around. So keep that in mind when you think about muscle fibers and specifically what their properties are. Now, I really like this table because it does a great job of succinctly showing the different characteristics of our different muscle fibers. So one thing I want to point out here is notice that our fastest muscle fibers are labeled type 2X. Sometimes in books that you'll see this written as a type 2B. In this case, they're referring to fibers that are found in animals, so often a rodent model, so something like a rat or a mouse. In humans, we only have type 2X, and this is just something that you'll notice between different textbooks if it's not human specific. So what are some of those characteristics that are really important that highlight the differences between our muscle fiber types? Well, firstly, our type 1 fibers are oxidative. So they're using oxidative metabolism to provide or utilize ATP. And so in this case, you need to have a high concentration of mitochondria to be producing the ATP for oxidative metabolism. So we do indeed see that in our type 1 fibers. They have very high proportions of mitochondria within them compared to our type 2A or our type 2X fibers. So because they're using oxidative metabolism, they're also very fatigue resistant, which is another important quality of these fibers. They do have slower myosin ATPase isoforms, meaning you won't see them contract as fast compared to our type 2 fibers, which makes sense. We tend to call them our slow fiber types. Now, when we think about our faster fiber types, our type 2 fibers, both our type 2A and our type 2X, remember, they just exist on a spectrum. Now they're utilizing glycolysis more as their form of metabolism, so producing ATP whenever sometimes we have some oxygen available, or in other cases we don't have any oxygen available. So they're not going to have as high a proportion of mitochondria within them, and instead they have very robustly developed sarcoplasmic reticulum because they're able to release a very quick and large bolus of calcium to the muscle to support very fast and very strong muscular contractions that are going to occur in these fiber types. They also have faster isoforms of myosin ATPase to allow the myosin head to ratchet more quickly, giving you that faster velocity of contraction. So make sure you spend some time making sure that you can go through and describe all the characteristics of our 3 different fiber types. And I tend to remember them just as type 1 and type 2, understanding that everything exists on that spectrum, and just that our type 2A are kind of that in-between intermediate fiber. So something I always wondered as a student was, how do we know what type of muscle fibers we're using or utilizing or that we have ourselves? And the only way to actually know that is to do a muscle biopsy. In the 1900s, a needle muscle biopsy procedure was developed, and it was actually specifically developed to be able to study muscular dystrophy. And then in the 1960s, we took that technique and we adapted it to be able to study exercise physiology and specifically what types of muscle fibers different athletes would have. So the process of actually doing a muscle biopsy, we would call that pretty minimally invasive. So I'll describe the process here. If you're a little woozy, maybe you don't listen to this part. But basically, you would take a local anesthetic once you decided where in the muscle or what part of the muscle you'd like to take the biopsy from, and a very small incision would be made. And from that small incision, they'd go through the skin, any subcutaneous tissue, as well as the connective tissue. And then you use what's called a hollow needle, and it's inserted to the appropriate depth within the muscle belly. And it has a little small plunger that is pushed through into the center of the needle to snip off just a very small sample of the muscle, which makes it pretty minimally invasive. That very little bit of muscle is cleaned and frozen, and then later it'll be thinly sliced, stained, and then examined under a microscope to be able to determine what type of fiber it is made of. Specifically, they're assessing for the type of myosin ATPase isoform that exists in the specific sample of muscle fiber that was taken. And you can use immunohistochemical staining or gel electrophoresis to then determine which type of myosin ATPase isoform you're looking at. This slide shows a really neat immunohistochemical staining of muscle fibers. And what's really neat about this is it shows you the different fiber types. So you'll see our slow fiber types are blue, or our type 1. Our intermediate fibers are green, and our type 2X fibers are in black. But what you'll notice on this figure is the red lines. And the red lines are showing you the dystrophin that's within the muscle. And the dystrophin is really important for holding that whole muscle together, providing it structure and allowing it to all shorten at the same time. And we're going to talk about muscle disorders, specifically muscular dystrophy, in which they don't have the dystrophin or there's a degradation of the dystrophin within the muscle that doesn't allow individuals with this disease to have appropriate muscular contractions. Now, related to sports, 2 of the predominant characteristics that we need to be concerned with for a muscle fiber or muscle fiber type has to do with its maximal shortening velocity, so the speed at which it can shorten those sarcomeres, as well as the maximal force that it can generate, which is going to be based on size. So if you look at these graphs, you'll see on the left-hand side that we have the shortening velocity, and type 1 are our slowest, and type 2X are our fastest in their ability to shorten. That same pattern holds true when we look at force in the upper right-hand corner. So our type 1 fibers that are our smaller fiber types, they create the smallest forces, and then our biggest fibers, our type 2X, create the strongest forces. Now, for most sports, what you're actually most concerned with is what is the maximal power, right? Because power factors in not only the force, but also the speed at which that force can be generated. For most sports, there's some sort of explosive component to those sports. So I have the example of a lineman in football, right? That needs to be a very explosive movement to be a successful lineman. And you can see that the same pattern holds true based on the other 2 independent characteristics, that the maximal power is generated by those type 2X fibers, and really the smallest, most minimal power is generated by our very tiny type 1 muscle fibers. So here's a fun question. Think about yourself, your own athletic abilities, maybe when you've played sports or trained in the past. If you had to guess, which fiber type do you think is most prevalent within your body, just based on genetics and how you've responded to training? I think one of the more interesting topics to reflect on is your own self as far as your past performance in sports and how you've been adaptable to different types of training. And this can tell you quite a bit about the type of fibers that make up your own body. Now, the average person has a pretty even split of about 50% slow fibers, so type 1, and about 50% fast fibers, so type 2A and type 2X in this case. Now, when we lay out these different types of athletes, we're really talking about the high-end elite athletes who have seen just a lot of success, the people you're going to see at the World Championships and at the Olympics and these types of events. So at a recreational level, yeah, you can be very successful with that 50-50 fiber type split, but if you want to make it to the Olympic start line, there's a good chance that you might have to have a high proportion of a certain type of muscle fiber to be successful enough to do that. So in our power athletes, things like our sprinters, they're going to have a very high percentage of fast twitch fibers. You see that on the bottom of this diagram where it represents about 80% of their fibers are fast fibers and about 20% are slow. In our endurance athletes, we see the opposite. So something like a distance runner, a marathon runner, we see about 85% of their fibers are slow fibers and about 15% are the fast fibers. So it's not only fiber type that determines success in an athletic event, but it certainly helps if you have the machinery to do the work that you're trying to do to be successful. I think for myself, I probably fall somewhere between speed skater and canoeist. So I wanted to show you this data that was recently done in American weightlifters. And it looked at world-class female weightlifters and then national level, both male and female weightlifters. And it showed what their fiber type distribution was. Something I want to point out here is that often sometimes people think that the type 2X fibers are going to be predominant. But what you see in this population, which is a hallmark of fast, explosive, very strong muscle fiber types, is actually it's those intermediate fibers, the type 2A, that are most prevalent. And this isn't uncommon to see in any sort of the sports where you have a very fast, strong contractions that are necessary. But just keep in mind, those type 2X, it's much more common that we just have a smaller proportion of those. And I think because we think of them on the spectrum and we don't always talk about quantity, it's maybe assumed that they're equal, or that if you do have predominantly type 2 fibers, that it's a lot of the type 2X. Not so much the case. Usually those type 2As will be predominant, especially in a highly trained population. So you should have covered this in applied anatomy, but we'll do just a quick review of the different types of muscular contractions that you have, because sometimes we use these in examples. You can have an isometric contraction, which means there's no change in length of the muscle. So a really good example here is when you're on the ground and doing a plank, that would be an isometric contraction. So there's no movement about any joint. Our other type of contractions are our dynamic contractions, or our isotonic contractions. And we have two different categories here. We have our concentric contractions and we have our eccentric contractions. So if I were to jump out into my garage and get up on our pull-up bar, and I were to try and pull myself up, that would be a concentric contraction of my bicep. So I'm shortening the muscle of the bicep as I'm going through the contraction. But if I were to help myself and assist myself up to the top of the pull-up bar and then slowly lower myself down, that would be an eccentric contraction in this case of my bicep. So it's still contracting, but it's lengthening in this case.